Hello and welcome. I'm Dr. Mark Brandon, an associate professor at the School of Medicine and the Department of Psychiatry at the Douglas Hospital Research Center, McGill University. As a neuroscientist, my research explores the intricate processes of memory, how we encode, process, and retrieve the vast array of experiences that form our lives. Today, I'm thrilled to welcome you to our publication series, a platform where I bring research from my lab directly to you, whether you're part of the scientific community or just intrigued by the inner workings of the brain. My mission is to unpack the complexities of neuroscience research and transform them into something accessible, engaging, and above all, inspiring. This episode focuses on our study from 2020, published in Nature Communications, titled DG-CA3 Circuitry Mediates Hippocampal Representations of Latent Information. Spearheaded by Dr. Alexandra Keinath, once a postdoctoral researcher in our group and now an assistant professor at the University of Illinois, Chicago, this research provides key insights into one of the most critical areas of our brains. Today, we're diving into how our brains encode memory. Specifically, we are looking at a fascinating part of the brain, the hippocampus. This area is like the control center for memory, helping us remember locations, events, and various environmental details. Picture this. The neurons in your hippocampus function as meticulous cartographers, mapping out your surroundings to help anchor your memories. For instance, consider how you might experience your kitchen differently, depending on whether you entered from the living room or come through the back door. Does your brain record this familiar space differently based on your point of entry? To explore this question, we designed a simple yet revealing experiment using mice. We tracked thousands of hippocampal neurons as these animals entered an open field arena from one of just two distinct entryways. Interestingly, we discovered that neurons indeed encoded the area differently based on the entryway used, much like how your perception of the kitchen might shift depending on where you're coming from. But how does this encoding take place? And what parts of the hippocampus are responsible for it? Our focus turned to the dentate, gyrus, and CA3 subregions, areas thought to play a major role in encoding and retrieving memories, especially in distinguishing between similar memories. To delve deeper, we repeated our experiments, but added a twist. We recorded from neurons in the CA1 region of the hippocampus while using chemogenetics to temporarily turn off the dentate, gyrus, and CA3 subnetworks. This technique allowed us to see what happens when we disrupt these specific parts of the hippocampus. The results were striking. Silencing the dentate, gyrus, and CA3 subregions erased distinctive hippocampal coding based on the animal's recent behavior, including which entryway it had used. Without the input from these two subregions, the neurons in CA1 stopped encoding these specific details, proving just how critical the DG-CA3 circuit is in managing memory based on our recent experiences. This isn't just a leap forward in understanding memory. It opens potential pathways for treating conditions that affect memory, such as Alzheimer's disease. By understanding these mechanisms, we can target therapies more effectively, potentially reshaping how the brain stores and recalls memories. Thank you for joining us today as we unpack the intricate details of how neural activity makes memory possible. Stay tuned for more insights into the remarkable capabilities of the brain in our upcoming publications.